Hello, I'm just going to start the first two tutorials. So the first tutorials that you will find in your build-through machines is about kind of an introduction to what BMTK, Sonata, and BND ecosystems are. So kind of why we make biorealistic models of brain processing. So the main idea behind this tool is to try to understand brain function, create like easy-to-use tools that make easier for scientists to create biorealistic models of the brain, to understand the relationship between local biological implementations of learning and abstract neural networks. Also, for understanding how different neural cell types contribute to different signals that we can record experimentally, but we don't have kind of the resolution for like kind of tests and manipulations of the things that are going on in the brain. And it gives you more understanding of how different excitability and different network components contribute to different pathologies. And learn how, for example, phenomena like oscillations emerge from different cell types or different network interactions. And very importantly, from humans, for example, we have EEG and MEG. And with this kind of biorealistic network, specifically biophysical networks, we can understand better how these signals and how different impairments in the signals come about, and gain a better understanding of brain functions and do manipulations that we cannot do experimentally, since we have these tools that allow us to do different level of resolutions and perturbations to our networks. For that, the Brain Model 2 kit was built. So this is a Python-based kit that facilitates the building, simulating, and analysis of large-scale brain networks at different levels of resolutions. So in BMTK, you can build biophysically detected networks that are also hybrid with point neuron networks, and you can build just point neural networks as well as filter net networks, which are usually used for creating realistic inputs in areas like LGM for our B1 network circuit, and you can also do population statistic networks. All of these done in a series of modulated steps. So first, you have a common builder or API for building all type of networks throughout the different level of resolutions that they have the same structure and the same functions. And then you use a series of simulators that are already widely used by the community and are kind of, I would say, kind of the gold standard for the different type of network resolutions. First, you have the API for using neuron with BMTK, which is called BioNet, which is for doing biorealistic neural models. And as in the B1, you can also combine point neurons with the biorealistic network. For doing just point neuron simulations, we use Nest. FilterNet, which is usually used for generating LGN responses to realistic visual stimuli, and now it has been expanded for auditory stimuli as well. We use FilterNet. And PubNet for population level statistics. Sorry about that. And all of these are stored and built in Sonata file format, and we also have a minimal set of tools for analyzing the simulated data. And we have we support common kind of file formats like NWB, which allows you to use the same analysis pipeline and other toolbox with Python and other softwares for analyzing your simulated data as you do with our experimental recorders and do one-to-one comparisons. BMTK allows for scalable to scale and parallelize your simulations without you having to do the kind of parallelize your code and all that. So it takes care of that for you, which facilitates your work. So you just have to worry about building your network. And the network is created and stored in Sonata file format, which uses already kind of build format. We don't create a new format for that. We use the ones available like HDF5 files, CSV, and JSON files. The main idea is that you store the information of the cell models, morphology, and other components in these formats, and as well as the network connectivity, external stimuli and the resulted activity in the same standardized format, which have a data structure. And you will have in H5 files the ones that are more complex connectivity information and the location of your cells, etc. And you have in more readable formats like JSON files and CSV, which are your cells, which kind of models are you using, and you can, after instantiating your model, are already built, and you can make modifications to those readable formats, and you don't have to repeat the building of the network, which is usually what takes most of the time. And is designed for store efficiently, user readability, and easy sharing across simulators as well as with other users. The main idea of this infrastructure is that you have an network builder. In this case, in the tutorial, we're using BMTK, but it's also supported for other software. And you create your Sonata circuit files that are stored in your network folder most of the time that have all the information of the connections of the cell types, the models, where are your synaptic components are stored, and all that information. And you create a separate JSON file with the configuration of your simulation. Where is your network files stored, how long is your simulation, where it starts. I will go into more detail in the second tutorial about this. And then you plug all this information into your simulation engine using BMTK, and you also save your results in Sonata file format. As for now, you can generate Sonata files in this set of simulators. So you could do your network in any of these and then upload your files, for example, to BMTK and run the same simulation since they are in this standardized format. And Sonata can also be important in these simulators if you want to use other tools as well. Yes? Yeah, so we use for the point neuron simulations Nest behind the hood using the BMTK API, but now you can also support directly using Nest the Sonata files as well. So the visual neuronal dynamics, or VND software, is used for visualizing, rendering, and animating models in the Sonata file format. So you can plot the location of the cells, the detailed morphology of the cells, and you can also visualize the connections between the cells. And you can check whether your cells are located properly in the network that you're building, the connections between them, is that what you want to do, and so on. So the main kind of model building workflow idea is that you build your network and then you import your files into BND and make sure that your connections, your cells are where you want them to be. It's kind of a way of checking. And then you run your simulation, and you can use also BND for visualizing the responses of the data, like the movie, the Anto show, but it also allows you to do raster plots and other tools that Barry will talk more about in the BND tutorial. So in this first tutorial, basically is for you to familiarize yourself with the tools, and also explains how to access the AWS instances. And ask Kel or any of us if you have any issues with working with the tutorials. And in the AWS instance, you have all the tutorials, including this one, and you can run them, but you can also have them in your computer as well from the GitHub repo. And always try to restart the kernel because it sometimes gives issues. I make sure that if you build the network several times, you delete your previous build network because you can get an error when you have already the build network or the output file. Sometimes if you don't specify to override your results, it won't override it for you. So here are a set of the main papers that presented these tools, as well as our BMTK repo, the documentation, and the BND website, and two other links for our layer 4 V1 model and the full mouse V1 model. So I will go over now the second tutorial, which is learning how to build a simple model Simple meaning one single cell in BMTK. Let me know if you have any questions along the way. So the first things that you need to choose is which cell model will you be using. For that, you have different options. As I mentioned, you can have point neural networks that will be running nests, or you can have conductance-based models that we use usually BioNet or Neuron. That's the tool that we're using in this tutorial. I will run a single cell biophysically detailed model in Neuron. And BMTK also allows you to use hub templates or models for examples as those that were previously developed that are available in ModelDB. There are optional tutorials that explain you how to import models that are not in the Sonata format into BMTK for running your simulations. And then you need to select your morphological model. We support SWC formats and ASC formats for the morphology. There is a tutorial 3A that give you more detail about the representation of the morphologies in BMTK and SWC files specifically. Usually we get the models from the Allen Cell Types database, which has thousands of models from the mouse and human cortex that are both biophysically detailed as well as point neuron models. For many of the cells, you have the 2 levels of resolution, so you can decide which kind of network model you want to do depending on your scientific question. You can also download models from Open Source Brain and ModelDB, as I mentioned. For this tutorial, I will explain how to download the models and use them for biophysically detailed perisomatic models in the cell type database. So if you go to the cell types database page, Allen Brain Atlas, that here is the website, you will see this interface where you can select whether you want human or mouse cells and specify the location. For example, here, primary visual cortex layer 4, and which type of, in the case of the mouse, transgenic line or cell type you want to download the model for. Here I selected GLIF model and perisomatic model, as I mentioned, will be mostly the ones that we use for these tutorials, but we also have all active dendritic models. For the perisomatic models is only the soma is active, so you have passive dendrites. And once you have selected these and filtered through the cell types that you want and the model types, you will see this kind of panel. And you can click in each of these and see the different properties of the cell, the electrophysiology, as well as the morphology. When you click in the electrophysiology, you will see this panel, where here on the top is the actual experimental results of the Pasch-Clan experiments, where you have the response of the cells, in this case, on their Lone square pulses for different current amplitudes. And these are the voltage traces that were recorded and the spike times. You can click here and you will have different type of stimuli, no stimuli, and so on. You can download this data as well. And when you select here in this portion, the model type, you will have several options, all the GLIF models that were fitted to that specific data, as well as the biophysically detailed models, which include perisomatic and all active. Here I selected the perisomatic, and it will pop up at the bottom the model with the specific model ID, and it will show you the response of the model for the same stimuli that you select for your experimental data. If you click here in the download button, you can download all the information of the model, meaning the JSON file, which is in Sonata format, that has all the information of the conductances of the model. You will have the SWC file with the morphology. We will use those files for importing and creating our network in BMTK. In this case, the network will just contain one single neuron, since it's a very simple one. So for building a single cell model in BMTK, we need to create nodes. In this case, in BMTK, nodes means cells, so it's kind of equivalent. And we create nodes for each of the different cell types. Here is a snippet of code that I will go into detail for each of the steps that kind of shows you how to create these nodes in BMTK. This is also in your notebook for tutorial number 2. And so you instantiate your network and start adding cells, which cells are the nodes. And in this case, we will have no synaptic connections. So the first example would be an iClamp experiment, which shows you stimulate your neuron with a lone square current pulse. And synaptic connections in BMTK are named edges, kind of like a graph. And in our network, we want to create one cell, no initial connections, and then save it in Sonata format into the network folder and use Bionet, which uses Neuron for running the simulation. So first, you instantiate the network using that you name cell. You can put whatever name you want to define your network as. This name will be the one that your files of the network would be named after. If you look at the network folder in the AWS machine. For that, you use the network builder class. This network builder class is the same whether you are creating a point neuron or a population level network or any of the options that you have in BMTK. Then you add the number of nodes with that specific type that you want to add to your populations. In this case, this is done with the add nodes function that have all of these parameters. Sorry, I went ahead of me. So the add nodes will allow you to add the number of cells with that specific type that you want to add, and a specific number of cells for that node is in the n variable, in this case just one cell. And then you have a set of parameters that define your node or cell type. If I want to have more than one cell, I will just increase the number of n, but all those cells within that specific configuration of the node will have the same biophysical or biophysical model description. So you have to specify for each biophysical model this set of attributes. The model type, you're telling BMTK that you are building a biophysical network model. The model template, in this case, usually for all the cells in the cell types database, you will use biophys1.hoc, which is the template in Neuron that it will use for creating your detailed biophysical model. The model processing here is perisomatic because we are using a perisomatic model. If you're using an all-active models, this changes. You have more detail in the tutorial. And the dynamic params is where your specifications of the biophysical model are. There you will have the conductance, which compartments of your model of sections are passive or active, and which ionic channels you're including in each with the maximum conductance, reversal potential. All those biophysical parameters are saved in this JSON file. That it has a specific instruction. You can take a look at it in the examples that are in your tutorial notebooks. And the morphology is the name of the morphology file within your components folders that I will explain more about it, that you will use for creating that specific biophysically detailed model. This portion here are a set of optional attributes. Yes?
So these 5 strings that we just declared, are all of these 5 strings referring to files that have to be pre-downloaded, or are they things that are set up in the package of the MTK? Which were the things that we had to prepare first?
So these ones were the ones that you should have downloaded in the first step from the cell types database. So these are external files. These are just kind of attributes, or you're telling BMTK to use specific functions for creating your model. So you don't need to download this. This will be kind of in your installation of BMTK. So this is just saying you will use biophysical models. So BMTK knows that you're using Bionet. Also, like in here, well, here it's not showing, but later in your simulation, you need to specify that you're using Bionet so that it uses Neuron and it doesn't use Nest for your simulation. This one here is to know which kind of hoc template you're using for discretizing your morphology and building your model in Neuron.
How do we know what the other options are for hoc templates?
For the Allen type database, it's just this one. If you download a new one, it will be different. And in the optional tutorial that says external models, it explains how to do it, which you need to create other functions with extra things. It's not that straightforward as in here because it's not built in the Sonata format. So it won't be this one, but this one will be the one that you use if you use an Allen model, basically. And the model processing, it would be I think the main two options are perisomatic and all active, which is whether you're using the perisomatic model from the LSI task database or the all active. You can create your own custom processing models. That is also I believe, in that same optional tutorial that I was mentioning. Kel, I believe, at some point, one of the tutorials will explain more about those. Otherwise, these are basically the options that we use for our own models. Does that answer your question? These other parameters or attributes from the nodes are optional, but they're very useful when you are connecting your cells with other cells in your network, or if you are creating virtual synaptic inputs to your network, because it allows you to filter out which cells you want, not only by the cell model, but also by their position, by the tuning angle, and other properties that you can add. These one are kind of the standard extra ones, the optional ones that you can add, but you can add any abstract parameter that you want to define for your network as well. It's very flexible. In this case, this one says that this is an excitatory neuron, that the model name, which can be anything that you want to name your cell. This one we use the name from the transgenic line of the cell type. The location of your cell, for example, if you're simulating, in this case is primary visual cortex, but if you're simulating LGN or you have virtual cells, this is where you define which portion of your entire model these nodes belong to, and also the layer. Other optional attributes that you can add is the location, XYZ location, the rotation angle, and other things that I believe in the next tutorials, which you have like a more complete network, not just a single set, you will see more about it. And finally, with the build and save functions, it will instantiate your network and save all the connections, all the properties that you define your model in the nodes and edges information that I will go now in the next slide for to explain the edges. For setting up the simulation environment, we have a function that is called create environment that will create most of these working directories. So for BMTK, we usually have this working directory where you have the component folder, that within that component folder you have the biophysical models, the morphologies, the mechanisms. In the case, if you have point neuron models, you will have another folder called point neuron models, and so on. So in your configuration, you will kind of say, tell BMTK to go to these folders to get the information it needs for building and simulating your network. When you run this create environment, you can put a path to the components folders, and you also name the configuration file that you want to create for the information of the network, where your network should be stored, the output directory of your network, the simulation length, as well as the discretization of your simulation. In this case, we're creating a current clamp, so you can specify here the amplitude of your current, the delay, and the duration of that stimulus. I will show in the next slide kind of what it looks like, the final configuration after you run this command. And you tell which kind of variables you want to store from your simulation. Generally, if you don't specify this, you will only store the spike times or your simulation. And here I'm telling that I want to store the membrane potential of all the cells, which will save the membrane potential of the sum of all the cells included in the network. And you tell BMTK to compile the mechanisms, because in this case you have mod files, same as in Neuron, for kind of the models of ionic currents, kinetics. And here it tells to overwrite the configuration. So in your case, I think in your folder you already have that configuration file created. If you want to create a new one because you updated this current clamp or other external inputs, you want to override that. Otherwise, you will run that and nothing will happen. And once you've run that, it will create this config iClamp JSON file, which have the information of your simulation, the inputs, where your network files are, and so on. And it will create a script called run Bayonet, which have all the code needed for running Neuron with BMTK. Yes. Oh. Yes.
Just sort of working through the network builder from the previous slide.
Mm-hmm.
Is this, I think I'm a little bit confused about this optional part.
Mm-hmm.
Do you, because I thought we downloaded that, all this info. Is it not?
It's in that when you download the model, it also contains this information.
Okay.
But it's in case you want to create like your own, or you want to create like, I don't know, nicknames or things to signal which node type. This comes more in handy when you are either analyzing or connected your cells to other cells. In this case, because I just have iClamp, it's like, it's there, but it's not doing anything yet.
Okay. Understood. Thank you.
You're welcome.
That is basically taking the strings of the group.
Could you, I couldn't.
So is that the functional part? Is it basically like tagging like strings?
Mm-hmm.
Attributes?
Yes, so here, yes, it's for the tagging, so that you know it's not a PV cell or something else. And it also tells you which layer. This is important for connections purposes. And also when you're plotting, even if you are just simulating like 1,000 unconnected cells, you can plot them with our analysis tools by layer. Sometimes here we also include like tuning angle or the orientation if you want to rotate your morphology and other kind of attributes that may use built-in BMTK functions that are processed when you run your model. But most of the time is for kind of knowing which cells to connect to which, whether they are close to each other, because you can add like X, Y, Z coordinates in your cortical column in there as well. Yes?
Question on the next slide. So when you specify the config file as the current clamp, that is specified as a current clamp at the SONA, and can you specify it anywhere in the morphology?
Anywhere. In this case, you create kind of the base JSON. After that, it's more practical. Well, it's better to do like your own modifications by hand, I would say, or create your own code for create a more sophisticated versions of your simulation. This we just create kind of the basic things you can do with that.
Sorry, another question. So
Yes.
when we have the report wires as the voltage, that's the somatic member information.
Yes, I will show more about that. That's why I was going to the next slide. So when you run that, this is what you get. A portion. I will go step by step of the different portions. So the first part is the manifest that tells you this is your working directory where all your folders that you will use will be kind of there. But you can also add like full path if you want to use like common components for different kind of networks that you have in different subfolders as well. The network directory is where you want your Sonata files of your network instance stored. Then you have your components. That is the folder here that has different subfolders. In the components folder, you can have templates, which is usually the BioFix one, hog template, or other templates that you use if you're using custom models, as in the optional tutorial. In there, you will have a folder for your synaptic models. In this case, what it means by synaptic models is like your time constants of the synapses and those kind of things that are also stored in JSON files. Usually we use the built-in kind of neuron exponential to synapse or alpha synapse sort of kind of things. But you can also create your own code for the synapses in Python. When you run it, it will use that. That is also explained in one of the optional tutorials. You have the biophysical neuron models, which these will contain all the JSONs of the biophysical parameters that you download for the cell type database. You have a folder for all the morphologies that you will use for your network. Another folder for the mechanisms, which contains all the mode files of your ionic channels. You have the filter models, which is for the filter net. So these folders will contain all the information, and you kind of combine different resolutions in here. And you have a folder for the point neuron models, if you're using lightness or leaf models in neuron. In the network section, you specify the nodes. You can have as many nodes as components you have in your network. In this case, because I just built a node with just one single network, and that called cell, you will have the cell node type CSV, which tells you the different type of model, detailed biophysical models that you're using. If I open this CSV, I will just see one cell, which is the SCNN1A model that I specified before. And it will also tell you what is the name of the file of your morphology. And all the other parameters that I show in the add nodes part will be there, specified in the CSV file. So if you want to change any of your optional attributes that are not mandatory by BMTK, you can change it in there after building. And you have in the H5 file, this we have all those parameters. For example, if you specify the XYZ coordinates, it would be in there. Like the node type, usually we use a number for like associated with each of the models that would be in there, as well as which node number is associated with which type of biophysical model. All that information is stored in there. The edges information we'll have once we build the synapses would be specified here, the path to those specific files. And if you have gap junctions that we won't use in this tutorial, it will also appear here. Other parts of the, yes?
For the templates, if you want to include a custom template, does it have to be Hope or could it be a Python?
It can be Python as well, yes. And we also support, I think, NeuroML and other that are also in the optional tutorials. You have way more detail about these ones, yeah. Usually in the main tutorials, we use the models from the cell type database. So in another portion of this configuration file, this is all inside the iClimb JSON file. You have the target simulator. This would be a neuron now because we're using BioNet. It will change to the other when you're doing another type of simulation. And under the run section, you specify your simulation parameters. When you want to start the simulation, for how long, the time discretization, in this case, the spatial discretization for your model when you build your model in Neuron, the spike threshold, as well as the steps that you will be saving your data. And you have the initial conditions for your simulation. And then you have the other part about the output files. In the create your environment, you saw that we specified the output folder. That information would be in the manifest, and here it shows the log, the name of your log file. We save all the information that you're running. All that you see in your terminal, it will be stored in that file, and it usually always creates this spike.h5 file that have all the spike times of all your cells. But on the reports, you can specify if you want to save further variables. For example, B report under membrane report is for saving membrane potential of the cells. And you can specify which section of the cell you want to record the membrane potential from. And you can specify which cells. So in here, it's useful the optional attributes that you added to your nodes portion, because you can specify if you want to just record from PV cells or excitatory cells as a whole, or all interneurons, or layer 4, certain type of cells. So all that can be specified here, and it's the same attributes that you use for your add nodes portion of your instantiation. And this is just the name of the variable when you're running the simulation. That's the name that the membrane potential takes in your simulation. Other variables that you can save here, for example, you can save the membrane potential of the synapse, or the intracellular calcium concentration, and kind of all the things that you can do in neuron or other simulations that you would like to have. You can specify those in here. And then there is another section that is called inputs. In the case of that I specify for created environment, I say that I want an iclam that had an amplitude of 150 picoamperes, that started 500 milliseconds after the beginning of the simulation for a total of 2 seconds. All that information is in here. You can change that after building your environment, because this is just in the JSON. You can put whatever you want. You can have several iclams, or add different type of current stimuli, like a sinusoidal and those kind of things. Even create your own current in Python, and then input here your result. And that is done with the module iclam. We also have like other modules for different inputs. That's also, I think there is another turn-up tutorial for that, for more kind of a specific type of stimuli for your network. And this is the result of that network that I built for this iclam. These are the spikes recorded at the summit of the neuron. You can create this plot with also built-in functions in BMTK. They are kind of basic for knowing if your simulation worked, which spikes you obtain, and those kind of things. Yes?
The current clamp here, when it's simulated in neuron, is the current clamp a predefined function, or it actually feeds in something like an NP Wi-Fi that says 0.15 for a whole bunch of time?
I think it's a built-in function that we have. I don't quite remember what it uses, sorry about that, but it's a built-in function, and it will have like an amplitude within that, but it's kind of, but you can also input like your own array if you want to have more specific things.
Say my own NP Wi-Fi. Let's say we're simulating this for 3 seconds at 0.1 second resolution. So if I want to specify my own 30,000 current values as NP Wi-Fi, is that possible?
I would think so, yes. And I also, you can, yes.
We will talk about it in optional
Yeah.
there's options for that.
But I believe so. And you can also, I was checking that yesterday, that you can specify like a series of currents and a series of a star and N. And you can add as many different sub-iclams for different cell types. So yeah, you have the flexibility of choosing which cell type, which model you want to simulate, and a series of different iclams. So in the second portion of the tutorial, you will learn how to add synaptic connections. In this case, we won't have more cells. We will have what we call virtual cells, which are kind of fictitious cells that activate your network, that are described by a spike train that is predefined by you. In this case, I will show a Poisson spike generator that we have built in. We also support, I think, gamma distributions for spike generation. And in the optional tutorial, you can see how to create your own kind of spike generation function from the ones that we have, the classes that we have for building those. So this is kind of the code that is in your notebook for creating that. We will use the main function for adding the connections to your cells, will be the add edges method. And you will have different properties and options for doing that. So I will go now to each portion of that. So first, as for the add nodes, you need to add the edges. In this case, we are adding the edges to the virtual populations that I don't have the code here, but it's the same procedure as before that you did for your biophysical model. In that case, instead of having biophysical, you will have virtual in your cell type. And you can see that specific code in your notebook. So you create the edges, so add your edges to your virtual cells, and then you need to specify which nodes are your pre-synaptic and your post-synaptic populations. For the sources is the pre-synaptic population, and the target is for your post-synaptic population. So here I'm saying that I will connect the virtual cells to the biophysical detail model that I built in the previous example. And then with the connection rule is where you specify how you want to connect your cells. You can have as elaborated connections rule as you want, like having into account the distance between the cells, the tuning angle, like in B1. But you can also have like a list of number of synapses that you want to have between the source populations, or just a single number. In this case, I use a single number. That means that we have 12 synaptic connections between that virtual population and the model that I'm building. So I will be activating the model with 12 synapses. And then here, similar to the add nodes, you will have to specify the model for the synapses. You have the model template. We have a set of built-in models that are the ones that are in neuron, like exponential 2 synapses. And in the dynamic parameters, which these JSON are stored in the components folder, you will have the information of the time constants of the synapses for this specific model type of synapse, and the reversal potential as well. And in here, you can specify the synaptic weights and the delay of that synapse connection. You can instantiate as many edges as the different models of synapses or properties that you want to add, because this one will make all the connections between these two the same. You cannot have different properties in the same instantiation. It's like a common property for all these connections. And you can filter out which kind of cells from this network, if I had more, you want to add. The next tutorial will explain more about that within this node folder. That's what I said, that the optional attributes in the nodes are important for when you are doing this, because you can filter out and select which kind of cells you want to connect to which. In this case, I just have one cell, so I just connected the virtual cell with 12 synapses to that specific cell. And you have a function called add properties that allows you to specify either have random connections from your pre-synaptic cell to your post-synaptic cell, or where in that biophysical detail model you want to locate your synapses, or whether you want to upload specific locations like XYZ coordinates of your synapses, and different options. For example, here we will specify the afferent section, which is the location within the postsynaptic neuron where you want these cells to be located, the ID of the section, as well as which postsynaptic section, whether it's the apical, the basal, or the soma of the cell. And for that, you can specify a rule. We will use the random synaptic locations rule from BMTK that's already built, which is kind of a random generator or selector of your afferent section ID and afferent section position, that it uses this set of parameters for getting those locations. So you specify which sections within your model you want the synapses to be located. In this case, just throughout the entire neuron. And you specify the distance range, which is the distance from the soma that you want those synapses. When you use this big number, it's just the entire neuron, but you can specify 50 microns, 100 and so on, or from 50 microns up to 300 microns from the soma. And it would be kind of the apical dendrites, and it won't include the soma or the basal dendrites of the cell. And this is just the morphology directory. By default, it assumes that your components are components morphologies is your path, but you can specify here if you have in an outside folder your morphologies. And as well as the discretization for the biophysical models for selecting those, because this will instantiate your neuron in the neuron simulation, so it needs to know like your discretization, so there is no discrepancy when you're running the simulation, so you're using the same one. And finally, after you specify all these, you also need to build your virtual cells and save them. This will create the other portion of the configuration file that has the AJ information. It will also create a CSV that has all the information of the synapses models that you're using, to which nodes, presynaptic sources, and targets. And it will also have a specific location of the synapses stored in the H5 file, and it will associate the source with the target, and all that is instantiated in that. Once you build all these, you don't need to rebuild it for running several simulations. You just need to change your configuration script if you want to run different kind of simulated experiments with the same network. This is an example of how to generate Poisson spike trains for your virtual cells using our built-in Python spike generator that you specify the node name, which will we use the virtual nodes, and you specify the fighting rates that will dictate how your virtual cell is fighting. This can be a number if you want a specific fixed fighting rate. In this case, we use a sinusoidal fighting rate, so it varies the fighting rate of the virtual cells and generates these 30 virtual spike trains, and this is just the histogram of spike times. And you can do this using this code and save it to Sonata format. Usually, we try to put all the external inputs into the inputs folder, but you can use whatever you want and just change the path in the configuration file. And we have like built-in plot raster functions that you can check what you're doing as you go on. Finally, you can check all these files using the HDF reader that is in the virtual machine, and you can check what I was mentioning, how these different properties are stored and the different structure of the Sonata files that you build in the network. And thank you. Let me know if you have any questions.